Hello, hi, a very happy good evening. So this is Krishna Ma'am and we'll be talking about the story of life on earth, and that's biology. So thank you so much for joining my session this evening. So please don't forget to like, share and subscribe. So today we are almost in the fourth lecture of photosynthesis. So we'll be talking about C3 and C4 plants. So that is our topic for today. So in the last class, I was teaching you what is cyclic photophosphorylation and what is 𝗘𝐴osmotic hypothesis. So we'll have a quick recap on the same and then move forward to talk about what is C3 and what is C4 cycle. Yes, ready? So I hope you remember the previous lecture. Yes, and also please give me suggestions on which chapter to start next. So according to me, I thought I will, after I finish photosynthesis, I would do biomolecules. So what is your opinion or suggestion? So please let me know. All right, so here we go forward to talk about photosynthesis in higher plants, lecture 4. Right, so I'll give you a quick recap of what we learned in the previous lecture. So probably I guess we have to do it with our hand. Yeah. Yes, we were done with all this, so I hope you remember, right? So we were done talking about a series of experiments. We also know where photosynthesis takes place. So how many pigments are involved in photosynthesis? So we have 4 pigments: chlorophyll A, B, xanthophylls, carotenoids. Then we know what is a light reaction, how an electron transport happens, and how in an electron transport chain ATP is getting released. What is a 𝐬𝐧 scheme? What is non-cyclic photophosphorylation? Where is splitting of water? And what happens in 𝗘𝐴osmotic hypothesis, right? Yes, ma'am, a very happy good evening. So thank you so much for joining my lecture this evening. So please do like, share and subscribe. Right, so what is the topic for today? So today the topic would be, where is this NADPH and ATPH used? So that is the topic for today. Yes, hi Rizwana Sayed, a very happy good evening. So I am able to recollect the message that you sent me in Telegram saying you like my lecture. So thank you so much. So hi Dinesh, hello, yes Pranam, thank you so much for joining my lecture today. So please do like, share and subscribe. So today we are in lecture 4 of photosynthesis. It's nice to see new people here. A very warm welcome. So thank you so much for watching my lecture. Hi W, yes, I was waiting for you. A very happy good evening. Yes, I'm good. How are you, W? So it seems like we are meeting after a pretty long time, though we are meeting after a day only. Yes? Okay, it's really nice to see you guys and interact with you. Okay, so we were done till 𝗘𝐴osmotic hypothesis in the previous class. So today we'll be talking about where this ATP and NADPH will be used. So we know as a product of 𝗘𝐴osmotic hypothesis, or as a product of light reaction, we have the ATP and NADPH. So where will this ATP and NADPH be used? How it will be used? In what form? So probably you can say, ma'am, ATP will be used to produce glucose, and the question is how? So that is what we are going to discuss today, right? So how it is getting fixed? So we are going to talk about who is going to accept your carbon dioxide and who is going to help in the production of glucose. So we have the very famous Calvin cycle, which is also known as the C3 cycle. Then you have the C4 cycle. That is what we are going to talk for today, right? So before we go to the C3 and the C4 cycle, so let me brief you about what I was talking in the previous lecture. So we start off with the light reaction. Next day, Menti, no, I will actually take one more lecture because today we'll do only C3 and C4. We still have C2 pathway and CAM pathway, which I will do it on Friday. Okay, so we also have to talk about the factors affecting photosynthesis. That I'll do on Friday. So Monday we will have Menti from this chapter. So according to my knowledge, I thought the next chapter that I should start should be biomolecules. If you guys have another idea, so please let me know well in advance. All right, yeah. So we know how light reactions happen and what sequence it happens in. So first, for the light reaction to happen, light has to be observed, point number 1. So point number 2, when light is observed or trapped by the L-HC, the electrons get excited, right? So when the electrons get excited, someone has to replace the electrons in its original place. So that is done by the splitting of water molecules. Once the electron moves forward, upward and downhill, and when it moves inside the electron transport chain, you have the ATP getting produced. So first step in light reaction, absorption of light. Second step, splitting of water. Third step, ATP formation. So now the electrons move towards the PS1, and after getting accepted by the primary electron acceptor, it reduces NAD into NADPH plus H+, right? So as a result of light reaction, so you have oxygen, which is a result of water splitting, and then you have ATP and NADPH produced, right? Fine. So then we went on to talk about what is cyclic non-phosphorylation and for non-cyclic photophosphorylation, right? So in case of your cyclic phosphorylation, so here only your PS1 is involved. So here only PS1 is involved and only ATP is produced. So how it happens? So light falls on the LHC. So the wavelength here is P700 nanometer. So it's accepted by the primary electron acceptor. So that is FES. But it does not go down. So it gets cycled back into the electron transport chain here. And then you have the plastocyanin, it comes again. So the electron keeps cycling inside the same concept. So that is why it is cyclic photophosphorylation and only PS1 is involved. So what happens in non-cyclic photophosphorylation? So non-cyclic photophosphorylation is also known as the ศ์scheme. Okay. So light falls on the LHC. So here the first photosystem that is active is PS2 and here the wavelength is P680 nanometer. So here the primary electron acceptor is your plastoquinone. So then it moves down into the electron transport system. So as the electrons move down, your ATP is getting produced from your ADP. Then it moves into the plastocyanin complex and then further into the LHC of the PS1. Right? So this is how it is happening. So here again the electrons will be accepted. So here the primary electron acceptor will be your iron sulfur complex. The electrons will move downhill. As it moves downhill, NADP will be reduced to NADPH plus H plus. So this is your ศ์scheme. So now to replenish the electrons that is getting excited here, so you have splitting of water molecules. So water molecule is split into its corresponding constituents of H2, O2 plus electrons. So water splitting is closely associated with PS2 and this oxygen is the byproduct of your photosynthesis. I hope you know the equation of photosynthesis. Yes. So you have carbon dioxide plus water giving you C6H12O6 plus oxygen. So I am not writing a balanced equation, I am just writing a non-stoichiometric equation, but this is the concept. So the oxygen comes from water, so now the carbon dioxide has to be converted into glucose, right? So this much is clear. Now when I said ATP is getting produced here, how is the ATP getting produced? So that was the concept of 𝗘𝐴osmotic hypothesis, right? Fine. So this 𝗘𝐴osmotic hypothesis suggests that for the formation of ATP, there should be H plus ions, right? So we are talking, I am recollecting your semi-osmotic hypothesis. So this 𝗘𝐴osmotic hypothesis, you can, it is, we are going to see how your ATP or your NADPH is being formed, right? So for this formation of ATP, you need a proton gradient. So gradient means difference in concentration. So we are going to talk about a condition. So imagine this is my thylakoid and this is my lumen of the thylakoid. And this entire portion is the stroma of the chloroplast. So we are going to form a gradient of H plus wherein the lumen has more H plus ion concentration and the stroma has less H plus ion concentration. Alright, so how come the lumen has more H plus ion concentration? So number one, the lumen has more H plus concentration by the splitting of water molecules. Right, so you have H plus ions here. Right, so this is also the reason you have more H plus concentration inside the lumen. So number two, it also helps in the reduction of NAD into NADPH plus H plus. So once again the protons get accumulated. The last reaction or the last method by which your protons are more inside the lumen is, so you have the ETC in your Z scheme. Yes, so ETC also needs electrons. The electrons grow from the stroma through a H plus carrier. The H plus carrier carries the electrons from the stroma to the lumen to the ETS of the lumen. So when the electron is delivered, the H plus also stays inside the lumen. So as a result, we have more concentration of H plus inside the lumen and less concentration in the stroma. Right? So once the concentration gradient is set up, it has to be broken. To break down the gradient, you have the F0 and the F1 of ATPase. So you have a component that is called as ATP synthase. So this ATP synthase has two components, F0 and F1. So F1 is peripheral membrane protein, F1 is integral membrane protein. So you'll understand more when I teach you biomolecules, alright. So when the lumen senses it has more H plus concentration, the H plus tries to move across the F0. When it moves across the F0, there's a conformational change in the F1, thus converting your ADP into ATP. So this is how your ATP is produced. Much in a similar way, your NADPH is produced. Thus, for the production of ATP, you need a membrane, right? So this happens across a membrane. So you need a membrane. Number two, you need a proton pump so that you have more concentration of proton. Then you need a proton gradient. And then at the end you need an ATP synthase complex. So these are the requirements for the production of ATP according to your chemiosmotic hypothesis. Now comes the next question. How is this ATP used in the production of glucose? So light reaction is done. So now the products of the light reaction are being used in the stroma for the production of sugar, and that is your dark reaction. So how in the dark reaction the ATP is being utilized comes the next question. Alright, so that is what we are going to see, and that is the topic for today. So where are the ATP and NADPH used? So the products of light reaction, so giving you an understanding, ATP, NADPH and oxygen, right? So you understood how oxygen is formed, how ATP and NADPH is formed, right? So we are going to the next part. So oxygen diffuses out of the chloroplast because it gets ready for respiration. So when we talk about the next chapter, that is respiration, which is in line in NCERT, you will understand how oxygen is being utilized there. So the fate of oxygen is that it goes into the mitochondria. Alright, so now the ATP and the NADPH, they are used to drive the processes that is in the synthesis of food, that is sugars. Alright. So it is used in the biosynthetic fate of photosynthesis or the dark reaction. So dark reaction is also known as the biosynthetic phase because that is where your sugar is being produced, right? So not directly dependent on light, but it is dependent on the products of light reaction. So it is in a way called as your dark reaction, alright? Now the question comes, how this reaction proceeded, or rather what was the first product that is formed when carbon dioxide is formed in the reaction or when it is phased. For example, say if I have gold and if I have to get a packet of biscuit for someone, so I cannot give the gold and take it, right? So first I have to exchange the gold to money, and from the money I can take a packet of biscuit, right? So now imagine I have carbon dioxide in the form of gold. Now I'll have to convert it in the form of money. Someone has to accept it and then give my end product, right? So who is going to do this? So who is going to accept the carbon dioxide first? And if they accept, what is the first stable compound formed? Because the final product is glucose. So in between there should be a product, right? So what is that? So how curious scientists were in those days, right? So with the way our brain works today, so I don't think we can find anything because our brains, I feel, have rusted during the lockdown days, right? Because we hardly studied, we hardly paid attention, right? Yeah, for people who are very smart, who are continuously working, it's working. But for the student community, I feel the efficiency of the working brain has come down with the advent of the lockdown, right? Fine. So here comes another scientist who's called as Melvin Calvin. So I agree, photosynthesis chapter has a volley of scientists' name, right? So many scientists' name you have. but yeah. So Melvin Calvin comes here, another scientist. So he used a radioactive carbon-14. It is an isotope of carbon. He used it in algal photosynthesis, and he found out that the first carbon fixation stable product is a 3-carbon compound. So when carbon dioxide is accepted by someone, it produces the first stable compound that is having a 3-carbon chain. So he called this as the C3 cycle, because 3-carbon is the first stable product. So that is called as the C3 cycle or the Calvin cycle. Alright, yes? Fine. So, okay, I'm sorry, this does not work. So the product that was formed or that he identified was phosphoglyceric acid, or in short we call it as the PGA. So this is the 3-carbon compound. Fine? Yes, understood? So moving further, so someone is accepting the carbon dioxide. So now carbon dioxide plus water So we know water gives oxygen. So someone has to work with the carbon dioxide to give us glucose. So Calvin said someone is accepting the carbon dioxide. We do not know yet whom. And the first stable product that is formed is a 3-carbon compound, that is the phosphoglyceric acid. So now in further steps it will be converted into glucose. So since the first stable compound formed is a 3-carbon compound, so this is called as the C3 cycle, or since it was discovered by Melvin Calvin, so we call it as the Calvin cycle. Fine? Yes. So that is the energy given by ATP molecule. Yes, W, you're right. Alright, fine. Now we are going to see who is that acceptor of carbon. Who is that willing to do that generous job? We are going to see. So the primary acceptor of carbon dioxide in your C3 cycle is a 5-carbon compound, that is RuBP. So RuBP stands for ribulose 1,3-bisphosphate. So this is what accepts your carbon dioxide and forms the first stable compound, that is phosphoglyceric acid, which is further reduced down into glucose. Alright, so this is your C3 cycle. Now there are certain plants, right, where the first stable compound formed is a 4-carbon compound. Say for example, let's take an example like this. In North India, you eat a lot of wheat. Right? In South India, you eat a lot of rice, right? So similarly, some plants, they form a stable 3-carbon compound. Some plants form a stable 4-carbon compound. That is the difference, right? It does not mean that the North Indian will never have rice, or neither the South Indian will never have atta, right? So it is much in the similar way. So in certain plants, the carbon dioxide is accepted by a compound X, and the stable compound formed is a 4-carbon compound, and since it forms a 4-carbon compound, this is called as the C4 cycle, or it was discovered by Hatch and Slack. Fine? Yes? So this stable 4-carbon compound is your oxaloacetic acid. We write it like this. Fine? So who is the acceptor here? So the acceptor in case of your C4 plant is your phosphoenol pyruvate. So your phosphoenol pyruvate is also a 3-carbon compound. So once again, you have two types of plants, C3 and C4. When carbon dioxide is getting accepted, if the first stable compound formed is a 3-carbon compound, that is your C3 cycle. If the first stable product formed is your 4-carbon compound, that is your C4 cycle. Right? So what is the C3 product that is formed? Phosphoglyceric acid. What is the C4 product that is formed? Oxaloacetic acid. Now, who accepts the carbon in both the cases? In C3 cycle, the primary acceptor is RuBP, ribulose 1,3-bisphosphate. Then you have In case of your C4 plant, it is phosphoenol pyruvate. So these are the acceptors. So you'll understand when we actually go into the process. So carbon dioxide assimilation is of two types, C3 and C4, right? So now we are going to see who is the primary acceptor, ribulose bisphosphate, alright? So now here we have the Calvin cycle. So here you have to understand, though you have C3 and C4, all green plants undergo C3 cycle. Irrespective of even if the first stable compound is your 4-carbon compound, they still undergo your C3 cycle. So we will see how C4 plants undergo C3 cycle very quickly. So as of today, I guess I can only do C3 cycle. So I will tell you how C3 cycle happens. Alright. So we'll directly go and I'll explain the C3 cycle. Right? So first step, carbon dioxide has to be accepted by ribulose bisphosphate, and it is getting converted into glyceraldehyde, phosphoglyceraldehyde, which is a 3-carbon compound. So this is a 5-carbon compound, right? So this step is step 1, and this is called as your carboxylation step. So who is the enzyme that is responsible? That is your Rubisco enzyme. Ribulose bisphosphate, carboxylase and oxygenase. Alright? Yes. C4 plants is only for a specific group of plants. Not all plants take up C4 pathway. Okay? Yes. Not all plants have C4 pathway. Neither all plants have C2 pathway. Only C3 is common. C4, C2, CAM are very, very specific. Alright, yes, okay, so this is carboxylation. So then your phosphoglyceric acid is reduced into C6H12O6. So this is reaction 2, this is reduction reaction. And during this reduction, so from 3 carbon to 6 carbon it is getting formed. So here you have to use 2 ATP. So 2 ATP and 2 NADPH is being used. So my question was how the ATP and NADPH is being used. This is how it is being used. Alright. Now the glucose molecule. So you have glucose molecule. Now it will undergo further reaction to regenerate the RUBP. Suppose your mom comes to drop you in school, your mom comes back home, she drops only you. Similarly, this RUBP which is being used here comes back again. This step is called as the regeneration step. This is step 3. And for regeneration, you need 1 molecule of ATP. So this is your C3 cycle. So first step is carboxylation, where carbon dioxide is fixed. For this we have 2 ATP. Okay. For this the enzyme is Rubisco. The second step is reduction, where the 3-carbon phosphoglyceric acid is converted into glucose, right? For which we have to use 2 ATP and 2 NADH. Then your RUBP is regenerated, so ATP is again used. So for 1 molecule of carbon dioxide in your C3 cycle, you use 2 plus 1, 3 ATP plus 2 NADH. But in the reaction or in the equation of photosynthesis, you have 6 molecules of carbon dioxide. So totally you have 18 ATP plus 12 NADPH being used. So this is used, not produced. Understood C3 cycle? Is it not easy? Is it tough? Now I will go to the previous slide where a lot of content was written. So step 1 is carboxylation, right? Fixation of carbon dioxide to form the stable organic intermediate. Crucial step of Calvin cycle because someone is accepting your carbon dioxide, right? The enzyme used is RUBP carboxylase, or you can also say Rubisco. So it forms the stable 3-phosphoglyceric acid, right? The enzyme is Rubisco. So now the next step is reduction. So it leads to a series of reduction steps to form glucose. For this we need 2 molecules of ATP and 2 molecules of NADPH, right? So the third step is regeneration, where RUBP is regenerated. So thus for 1 molecule of glucose, you need 6 molecules of carbon dioxide. So crucial means important. Crucial means very important, alright. So for example, the day before your exam is a very crucial night. A month before exam is very crucial. It is very important, right? So that is what I mean. So for 6 molecules of carbon dioxide, which gives us 1 molecule of glucose, you need 18 ATP and 12 NADPH, alright? Yes? Understood your C3 cycle? Was it easy? I don't think it was tough. Was it? Clear all of you? So I will do C4 cycle and C2 cycle on Friday. So give me one more lecture, alright? So yeah, so we'll stop here. We'll continue on Friday. So I hope you have understood where your ATP is used and how it is getting fixed, alright? So thank you so much for joining my lecture. So I hope you enjoyed. So please read corresponding NCERT portion and please solve a lot of questions. That is very important. So I will meet you on Friday. So until then, this is me signing off, and please don't forget to like, share and subscribe and refer your friend to this channel. So thank you once again and have a great evening. Bye.